hope you're enjoying our meeting. And we are delighted to welcome you today for this session. Our session today is, well, I can't remember it by name. Let me see here. I'm sorry for that. Biological invasions in shallow lakes. I want to be precise. Well, I'm just introducing the session, but the coordinators are Dr. Bruno Figueiredo and Roger Paulo. So you have the word. It was just a brief introduction. So whatever and whenever you need, I'll be here. So that's it.
Okay, thanks, Rosenberg. It's a great pleasure to be here. So it's quite complicated to talk about biological invasions after such a complete lecture by Dr. Sidney Thomas. But okay. But hi, I'm Bruno Figueiredo, a Brazilian professor at Federal University of Santa Catarina. And I must say, I'm so glad to be here to coordinate these thematic sessions in Shallow Lakes Conference with many of my references and colleagues on the audience.
Okay, bonito.
Me and my dear old friend, Dr. Roger, are the coordinators of these thematic sessions, named Biological Invasions in Shallow Lakes. Biological invasion is a subject that we have studied and contributed with it, mainly inspired by our masters, Dr. Sidney Magel. I will share with you my screen to show my presentation. Okay. Are everybody seeing my screen? Okay, perfect. So the proposal of these sections will deal with biological invasions in different ecological levels and organizations. Also considering a variety of concepts related to establishment and the persistence of invasive species in shallow lakes. The topic is quite timely. Sorry, okay, the coordinators, me and Roger with his family. And the topic is quite timely because human activities and globalizations has removed many species dispersal barriers, increasing the chance of an invasive species to arrive in a new area. And in invaded areas, invasive species may have catastrophic impacts on different ecological levels throughout the time, impacting the biodiversity, the ecology of invaded areas. For example, as shown in this figure, the arrival of a new species, and the establishment and the spread. In this other figure, we can see the homogenizations, one of the consequences from biological invasions. We received 9 quite nice and diverse contributions, 3 of them including the study of aquatic macrophytes, Elodia nutali, Urucloa arrecta, and Progmites australis, and 6 of them including animal species, invertebrate ones and vertebrate ones. And these contributions, including covered a lot of topics in ecology, some like how are biological traits of invasive species in invaded areas, or how do invasive species affect the native ones? These include the community response, ecological ecosystem response. Other questions, how are native and invasive species impacted by pollutants? How does an invasive species interact with another? And what is the impact of invasive species on the nutrient availability? So it's a huge of ecological levels and organizations being impacted by invasive species. So I would like to invite the authors of these contributions to present a quite brief summary of their abstract, and then we can ask each one to, anyway, the questions you have. So the beginning is Ms. Leticia Escalante, or some other author of this work,
Hello.
Hello, Leticia.
Can I share my screen?
Okay.
Here it is. Thank you. Well, hello everyone. My name is Leticia Cruz. I am from Mexico. I have just started my PhD studies at the National Autonomous University of Mexico. My work is titled Acute Toxicity Effect of Copper on Freshwater Copepods, Mesocyclops pepayensis and Mesocyclops longicetus. Well, as you may watch in my video, water pollution by copper and invasive species is a huge problem in Mexico. Mesocyclops pepayensis, marked with green, it's an invasive species from Asia that has spread in Mexico since 2013, when it was first reported in Chiapas in the southeast part of Mexico by Suarez Morales et al. On the other hand, Mesocyclops longicetus is native of America. It is naturally distributed in Tabasco, Veracruz, Aguascalientes, among other Mexican states. Both species are efficient predators and are used as control of mosquito larvae. Having in mind this problem and the fact that freshwater copepods are not used very often as bioassay organisms in ecotoxicological tests, sensitive to copper, and if there are any difference between the sensitivity of an invasive and a native species. So we perform an acute toxicity test using adult females at 24 centigrades with 5 different concentrations of copper. The concentrations that we used, or my treatments, were 40, 42, 44, 46, and 48 milligrams per liter of copper. We found out that all organisms survived at 40 milligrams per liter. Mortality was observed at 42, 44, 46, and 48 milligrams per liter. The sensitivity of both species was similar, 1.6 females of Mesocyclops pepayensis versus 2 females of Mesocyclops longicetus died on average at the 42 milligrams per liter treatment. Both species were less sensitive to copper compared with other freshwater copepods already tested in Mexico, such as Acanthocyclops americanus, tested by Sabino Figueroa. We will conduct, these are preliminary results. We will conduct further tests to clarify the sensitivity of each species. Thank you.
Thank you, Leticia. So the questions for the work of Leticia. Please use the tool, raise up your hands to better organization. Anyone? I have one question. This experiment included 6 concentrations of copper, yeah? And are such concentrations comparable to levels found in nature, or they are just comparable to sites where human-mediated copper inputs occurred?
Now they're based in works and in natural concentrations in Mexico. We have concentrations that goes from 2 milligrams per liter up to 90 milligrams per liter in some places due to minery or something like that. The example that I used in my video about the accidental spill, well, when that happened in 2013, they found up to 300 milligrams per liter of copper diluted in water bodies. And that was an ecological disaster that is still affecting water bodies today.
Okay, thanks. Thank you.
May I ask a question? For Daphnia ecotoxicology test, they usually use neonates. And why do you decide to use adults? And how do you deal with sexual differences because copepods are breeding, are sexual?
Well, I will use neonates as well, but that will be like another step of my work. And I didn't understand the question about the sexual. What
Yeah, and on rotifers and Daphnia, they are parthenogenetic organisms, and you don't have a genetic difference that affect your results. Copepods are sexual, and you can differ genetic differences between or among individuals. So I don't know if you think that it could affect your results.
I'm not sure about it, really. I have not considered that genetic part. I'm trying to focus on the aspect of invasive organisms and invasive species already established in a system and try to compare it with a native one. Because, you know, there are like two parts of invasion. That one says that the native species is used to the environment and that it has the advantage of survival, because when the when the invasive species came, it is not used to the environment. So maybe the invasive species has some problem to be there because of its nature. And there's another part that says that invasive species are quite resistant to a lot of factors, and that's why they are spreading. So that's more my point of view, that I want to compare that an established organisms and established population, how do an invasive and a native species react in a system. And in Mexico, at least, most of our systems are polluted. So in laboratory conditions, maybe they can show some behavior, but with different factor stressors, multiple stressors, maybe they could be different. So I haven't taken the genetic part, but it could be really interesting.
Okay. Thank you.
Okay, no one more question or comment? Not only questions. But okay, thanks, Leticia. We can move on for another presentation.
Thank you.
Thank you very much. The next presenting is Dr. Sabine Hilch. And I can share my screen, or if you want.
Yeah, hello everybody. Also, thank you for the organizers to organize it in this nice way. So my topic was the mutual facilitation and boom-bust dynamics of invaders, which seem to pave the way for a native species. And in my, if you know me, in my case, it's about macrophytes mainly. So you may know that in Berlin, Germany, we have a long-term program running in a polymictic shallow lake in front of our institute since more than 40 years. And those long-term programs are, as usual, focused on planktonic organisms quite a lot. But some years ago, luckily, we also started looking at benthic parts. And in around 2013, we realized that all of a sudden the water clarity increased quite a lot. And it seemed that something has changed in that lake very dramatically. And we hypothesized that these clear water conditions were both the cause and the consequence of a mutual facilitation among an invasive mussel, Dreissena rostriformis bugensis, and the invasive macrophyte Evidea nutali. Then we also hypothesized that Elodea nutali colonization follows a so-called boom-bust dynamics, so a sudden rise of a population and followed by a dramatic decline. These species, Elodea species, are known for such a dynamics. And thirdly, we hypothesized that native macrophytes would subsequently then gain from this mutual facilitation and boom-bust dynamics and also increase in abundance. Next, please. So here, these results, in fact, you can already read in a paper. It's Wegener et al. 2019 in Frontiers of Plant Sciences, where we described this initial mutual facilitation among Elodea and Dreissena rostriformis bugensis, which on the one hand side, the plants provide surface for the younger mussels to attach to, and on the other hand side, the mussels provide by clearing up the water, new habitat in the deeper parts of the lake for the Elodea, which can easily colonize, and then also additional carbon dioxide and phosphorus also for the macrophytes. So we found a nice correlation between actually the quantity of the dreissenids, the quagga mussels, and the macrophytes, but only for Elodea and only until 2018. And then what is not in that paper is the subsequent development of the macrophyte. So we had a takeover of the macrophyte community by Elodea in 2 years between 2016 and 17 was the peak, and then 18 and 19 was dropping, and in 20 species was almost gone. So we had a really nice, very typical development of Elodea with a strong boom and a strong bust in altogether 4 years. But still, as you can see from these blue bars here, these are the maximum colonization depths of macrophytes. So these remained high, and what happened was that native species took over and gained in dominance, and that's mainly species that have a certain number of traits that seem to help them in taking advantages of these new habitats, which in our case was Nias major and Certophyllum demersum. Next, please. So in the end, what we found in that little scheme here was this, that we initially had this mutual facilitation among quagga and macrophytes that's described in that paper. Then in the following 2 years, we had this typical bust of Elodea nutali. And then, but we didn't have a decline in macrophytes, but instead we had a further increase in native macrophytes, which in turn also, of course, provides surface for the quagga mussels. So there is no reason to assume that those mussels would decline again. And that was quite amazing how they took over, because we did have the other Dreissena species, zebra mussels, in the lake for already 100 years, but they could never have the same impact because they are restricted to the shallower areas with some hard substrates, while these quagga mussels can form a carpet, they can stick to each other, and they can colonize almost the entire lake. So in our lake, they are present down to 5 or 6 meters depth, and that's why they can have a pretty different impact here. And now they are even more supported than by the both native and invasive macrophytes. So that was it. And in fact, what we still don't know is why the Elodea nutali was declining again. So there is some speculation about rat, which is the only herbivorous fish species in the lake, which could have contributed. There's a slight increase visible. We have fish data, but it has been described in other lakes before that this may be the reason for the bust. But what we still need for that species is more case studies. So that would also be a call for, if people do have long-term data on Elodea boom-bust dynamics, I would be more than happy to receive them and to collect them. And then I think by doing so, having a whole lot of examples from different lakes, trying to find out what could make this species declining after this initial bust phase. And yeah, so please contact me if you have or know of such cases, and I'll try to work on that in the coming years. Thank you.
Thank you, Sabine. Christian.
Yeah, very nice presentation. And I would like to know if you think that allelopathy from Ceratophyllum or other native macrophytes can be involved in the reduction of Phyllodia.
I would doubt so, to be honest, because Elodea itself also produces these polyphenols. So I think all of them are quite strong in the production of these allelochemicals. And maybe if you do lab experiments, you could find such an interaction, but I would, in practice, in the field, I would doubt that that would be a major mechanism leading to the disappearance of Elodea. And the interesting thing is that with the Elodea natalis, there is lakes where there is no bust, so they can persist for decades. We have examples of lakes where they have to put a lot of money to mow them every year, and still even that mowing doesn't lead to the takeover of other native species. So it's quite amazing how some lakes have that bust and others don't. And yeah, so possible, but it's just a gut feeling. My gut feeling says might not be the major reason, but who knows? Maybe.
Thank you.
Rosenberg, raise your hand.
Yes, thank you very much for your very nice presentation, Sabine. Actually, I have a curiosity about your work. I was wondering if this system somehow will become a desert in the future, since there is a decrease in the phytoplankton productivity, I suppose. And then, well, phytoplankton is very important. I mean, it's at the bottom of the food chain. So this may affect the top of the food web. I don't know if this lake has fish, or I don't know the size of the lake as well, but I was just wondering if this could become a local pattern, let's say, because of the introduction of Dreisena.
Yeah, thanks for the question. Quite interesting, but I wouldn't expect that, because in principle, what this water clarity leads to is new open spaces for other organisms, like, for instance, also the macrophytes. And if you compare lakes with like clear water and a lot of benthic organisms versus these turbid phytoplankton dominated lakes, then you often find a higher diversity. So in that respect, I would not expect that overall, just because of the filtration capacity of the mussels, that we have a desertification, because in principle the clear water just opens new niches for benthic organisms, which can be quite rich. So till here, I think till now, we have good evidence for a highly diverse space in the benthic, which is not restricted by the mussels. So I wouldn't expect that.
Thank you.
Fish production wise, there could be a decline, but that doesn't mean a desertification. I mean, we still are, in terms of nutrient loading, we are still above any natural background situation. So we just, anyway, produce too much. So I think that we've been through that desert phase when the lake has been plankton dominated for 20 years between 1970 and 1990. So you could have called this a desert, but it depends on your perspective, of course.
Hi, Eugene.
Whether the quagga is a good thing in this case. The thing is, of course, in terms of it was the disturbance that was needed actually for that lake to make a big jump in terms of macrophytes, because I've been following those macrophytes the last 20 years, actually, all my career here at this institute, and it took more than 10 years to reach a few more species. Initially, it was just Potamogeton pectinatus down to 1 meter, and that was it. So although the nutrient loading was already reduced quite substantially since 20 years, it took forever for the macrophytes to recolonize. So it needed a kind of extra disturbance to reach these new habitats and to colonize them. So in that respect, it was a good thing, but again, it depends on your perspective, because of course these quagga are also quite detrimental. I mean, they have been investigated also for their negative effects on like native mussels and so forth. So, but for the overall system diversity, my guess would be the overall effect was positive in this case.
Eugene?
Yeah, thank you, Sabina, for the nice talk. I'm wondering, like in the initial stage when their mussels they begin to invade their lake, for what I know, they mainly filter the water, then they can increase their water clarity. But I think for their lake mucosae, due to their mussels, they can spread from like 1 meter to kind of like to very deeper part, like 6 meter. But what I saw is that the mussel beds, they also can work as a substrate for the perphyton growth. Did you also quantify like how this mussel beds they work as a substrate to support the growth of the perphyton, then to facilitate their water clarity?
Yeah, I think that's a nice point. They do so, but I think because they are 3D, but they are still, the increase in surface is limited as compared to the increase in surface by the new macrophytes that also increased in their depth. So, but I have never calculated and compared what provided more new surface, the extra mussels or the extra macrophytes. So maybe that's a good point to do so. But I think the mussels as compared to a bare sediment surface are maybe factor, I don't know, 2, 3, 4, something like that. And macrophyte is maybe factor 10 something. So, but yeah, good. It's a nice point. What was also, by the way, interesting that we saw all these effects that have been described in the Great Lakes regarding the transformation of phosphorus from the plankton towards the more littoral shoreline area. So we saw like filamentous algae entangled among the macrophytes. So all these effects that have been described for other lakes, we even find in eutrophic lakes, although you would expect they have never been phosphorus limited. We are still in the phosphorus concentration quite high, but still this concentrating effect and then bringing phosphorus from the pelagic to the bendic area still had this extreme effect on Cladophora mass development in some years. Quite extreme.
Okay, great, thanks.
Okay, I think we have time for one last question, if anyone has. No questions. So thank you, Sabine, for your presentation. A nice contribution. We now move forward to the next presenter. Bruno, please.
Hi.
Hi, Fabielle.
So, in this study, used a large database, more than 3,000 samples, to investigate how an invasive macrophyte, the Urocoa recta, affected the biodiversity of native macrophytes assemblies over 13 years. For this, we calculated the biodiversity of native macrophytes assemblies with and without the invasive species to investigate spatial dissimilarity. In addition, we also calculated the local contribution to biodiversity of these assemblies with and without invasive species over time. We evaluated the biodiversity for the overall macrophytes assemblies and the 3 functional groups, life forms of aquatic macrophytes. So our results showed that the presence of the invasive species became the macrophytes assemblies more homogeneous in both space and over time. Also, there was loss of species within the functional groups, decreasing the functional redundancy of the assemblies over time. So that's it. Thank you.
Okay, thank you very much, Fabielle.
Thank you.
We are open to questions now. Come on, guys. Nobody, don't be shy. If you are too shy, you can write your question in the chat.
Hi, good morning.
Morning, Tal.
Yes, I would like to know about the physical and chemical characteristics. First of all, it's a huge study with a lot of data, so thank you to share with us. But I would like to know about that homogenization, but in the species, but also by the traits. But what happened with the physical and chemical characteristics? If something interesting to say about that.
Thank you for the physical and chemical characteristics for. Sorry, I'm nervous.
Don't worry.
Measure the physical and chemical, but in this work, not the additional these characteristics.
Did not change throughout the time. I can't remember. The characteristics did not change over the time because
yes,
between the presence and the absence of the invasive species. I can't remember.
Sorry.
The pH, the limnological characteristics differ between the presence and the absence of invasive species. I can't remember. Roger, do you know about that?
About the changes, please. So, could you please repeat the question for us?
No, I would like to know if, because you said that homogenization about the traits in the species,
yep.
But if you see something interesting about these characteristics that sometimes is the filter for that traits. I don't know if I could you could answer to me later. Don't worry. All are nervous.
Yeah, but we have a clear pattern in the reservoir of change in the environmental variables. For example, when we take samples far from the reservoir or in the beginning of the reservoir, the lake of the reservoir, we find, for example, high concentration of phosphorus and nitrogen and turbid water, and we can especially find floating plants, while when we take samples close to the dam, in the deeper area of the reservoir, the water is really clear and oligotrophic, and we find most of the submerged species. So we have a special spatial pattern of trait distribution and biological forms, and this is intensified through time. So the main invasive species is the submerged species Hydrilla verticillata, and also the Urochloa ahecta, that is an emergent species. So these species colonize fast the places and change the community composition, and also homogenize the traits of the species, the native species. But I think it is not only related to the homogenization is not only related to the presence of the non-native species, but also related to these environmental variables that predominate due to the, how can I say, it is like it's like zonation. You have different zones that we have different domination of some variables. For example, nutrients far from the reservoir and water clarity close to the reservoir. So we also have this effect. I'm not sure if I answered your question clearly.
It's okay. Thank you very much.
Okay, it seems we have another question in the chat. I think this is a very controversial topic. Invasive to be good. It will maybe need a temporal context to invasive species impact on appropriate measurement of indirect impacts. We'd love to hear from others present about it. I think this question is to Fabielli, Rafael.
No, I was just answering Annette Jensen. She wrote in the chat that a question for the previous presentation.
Okay.
We certainly discussed this topic in my next abstract from Maria Beatrice Castelliani.
I think your sound is turned off. I think you are mute. We cannot hear you.
Me? Okay.
No, that's okay. We are.
I can hear Bruno.
Yeah. Okay, thanks. Rosenberg, you're wrong. So in another abstract, we will discuss this topic, these questions. Are invasive species to be a good thing?
Yeah, that's a long discussion. Okay. Someone have more questions to Fabielli? Okay, if we have no more questions to Fabielli, we move on because we have not too much time. Let's see.
Dr. Sarma has one.
Yeah, I just may like to make some clarification.
Yeah.
I don't know if you can hear. It is important to distinguish between exotic species and invasive species. Okay, exotic species means that species that should not be there, but it is there, but doesn't cause any change in the local community structure. Invasive species, by definition, it is an exotic species in the beginning, but causes a lot of change in the local community structure, including displacement of the local species, sensitive local species. Now, the question that is, invasive species, are they good? Invasive species are never good. But what is good is exotic species. Exotic species, some of the exotic species are to be treated as invited species. Every country has exotic species because they have certain purpose, economic purpose mainly in every country. But we do not, no country wants an invasive species because that dislodges local species. Now, exotic species, if not controlled, over time becomes invasive species. That is the definition. So I would like to consider this point. This is only a comment, not a question. Okay, thank you.
It's a good point, Dr. Sarma. The problem is recognize the exotic species that may become an invasive one, no?
Yes, that is why you need to study before and after.
Yeah. Perfect. Okay, let's move on to the next presentation. Okay, I invite Laura Harkonnen to present his manuscript. She's there? She's here or not? I think no. Okay.
Bruno, sorry, but Laura sent the email because she didn't enter in the Zoom.
Oh, dear. I'm sorry.
I don't know why.
So for passcode, but I can't find one. Could someone please tell me the code? I'm so sorry. Okay, I cannot. I don't know answer her, but okay. Let's move on, and I try to invite her after. Thanks, Fabielle. Okay, the next one is the Maria Beatrice Castelliani.
Good morning, everybody. Can you hear me?
Yes.
Please, can you share the slide, please?
Okay. 1 second.
Yes.
Okay.
Good morning. The work presented here has three main goals: evaluating if there were differences in alien species between lentic and lotic habitats, evaluating the effect of habitat, alien species, and their interaction on taxonomic, functional, and phylogenetic diversity of plant communities dominated by Phragmites australis, and finally, evaluating the effect of habitat, status of invasion, and their interaction term on the different features of diversity, comparing the results obtained, included or not including the alien species in the computation of the indices. Please, can you skip to the other slide? Looking at results, we found that lotic habitats show a greater relative abundance of alien species .
Maria is muted.
Okay, sorry. Where I start again? Oh.
Please start this slide, please. I'm
okay,
sorry.
This slide. Sorry. Looking to the results, we found that lotic habitats show a greater relative abundance of alien species than lentic habitats. Regarding the incidence of alien species, we saw that the alien species richness increased taxonomic, phylogenetic, and functional diversity in lentic habitats, suggesting a possible presence of a changing enrichment structure. On the other hand, lotic habitats show a normal trends of open and dynamic environments subjected to species turnover. Please, can you skip? And at last, we found that the higher taxonomic, phylogenetic, and functional diversity within invaded plots is given by the alien species. In conclusion, thanks to this work, we had a better understanding of plant diversity between reeds of lentic and lotic habitats, and of the incidence of alien plants on taxonomic, functional, and phylogenetic diversity on these particular communities. Thank you. So if there are any questions, I don't know.
I have one question. I may have misunderstood, but can we consider the the occurrence of invasive species positively affect the native community are quite unexpected, no? Naturally, the interpretation should be cautious, as other potential negative effects were not included in the analysis, such as, for example, ecosystems functioning, ecosystem services. Do you believe that including time as a factor in the models, you would see a different trend, positively or negatively? What do you think about that?
Yes, of course, the time is one of the factors we can choose, we can take in account. But for me, the main factor we can take in account is the scale that we use for our investigation, because we probably have different results if we study a community at regional scales or global ecosystem scales. So in my works, I use is not as the smaller scale, but it's not the ecosystem scale. So it's only for the Italy, for the central, and for the north. So probably if I use for my analysis also the macrophytes communities to the south of Italy, probably the results could change, because Italy is very different in all the latitudes of the region. So yes, and for the effect of the invasive species, in my case, I say that we have a positive, statistical positive effect on the functional, phylogenetic, and taxonomic diversity. What I mean that other invasive alien species that arrive in our ecosystems are phylogenetic different from the native, of course, and they have other functional traits that are different from the native one. So if we can accept this like a positive way from the alien species, I don't know, but yes, we have to see in the time what happens after the alien species arrived in the community. Probably after 100 years, the situation is not better. Will be not better.
Any more questions?
I have another comment, and just a comment, not a criticism. But in the conclusions, you said the higher taxonomic, phylogenetic, and functional diversity within invaded plots is not given by the alien species, but linked to a great environmental dynamics.
Yeah, yeah. No, I'm apologize because the sentence in the third conclusion is not true. I make a mistake to write during the so I'm very sorry, but I saw in the second moment and it was impossible to change. So I'm very sorry, but no, it is from the alien species and the effect in the invasion plot.
Oh, perfect.
Sorry.
Okay, if we have no more questions, we have time to, oh, wow. Let's see here, we have a chat. Were these Atlantic habitats that had an increase in functional, phylogenetic, and taxonomic diversity with the presence of the alien macrophyte homogeneous environments? Did you see the chat, Maria?
Yes, I'm reading.
Okay.
So if I understand the question, the habitats are all lakes. So yes, are homogeneous environments.
Okay.
every habitat are really standard, our community. Sorry, okay. All the habitats are lakes or rivers dominated by Phragmites australis. So we have habitats with a cover of Phragmites australis over the 50%.
Okay, if you have no more questions, thank you, Maria.
Thank you
for your work. Please, Bruno. Who is the next?
The next is Dr. Vargas, Miss Litzley Vargas. She's here.
Hi. Yes, I'm here. Do you hear me?
Okay. Okay.
Yeah. Okay. Hello, everyone. My name is Metzli, and I am from Mexico. And I am working with the invasive species Mesocyclus pepeiensis and the interaction with the native species Mesocyclus longicetus curvatus, focus on their feeding behavior. In Mexico, we have 485 invasive species. Of these, 90 are freshwater species. About Copepoda group, in Mexico, we have 4 invasive species. One of these is my target species, the Chinese Mesocyclus pepeiensis, who has been found so far in 3 different Mexican states. In 13, we can say that it has had a fast dispersal. Mesocyclus pepeiensis is distributed in the world. It has been reported in 14 countries, colonizing different environments such as artificial water containers, hypersaline lakes, and warm water deposits, and others. It has been shown by different authors that Mesocyclus pepeiensis have several attributes of an aquatic invasive species, for example, a short generation capacity and a broad diet, and wide environment tolerance, and among others. So in this work, we wanted to know if Mesocyclus pepeiensis is capable to displace native species like Mesocyclus longicetus curvatus. The next one, please. Therefore, evaluate the resource consumption efficiency of the invasive Copepod Mesocyclus pepeiensis over the native Copepod Mesocyclus longicetus curvatus based on their consumption rates and food preference, separately and together. The results, the consumption rates show that we can see on the top in the first graph, in separate environments, Mesocyclus pepeiensis had a total consumption of 7 individuals. While in the second graph, Mesocyclus longicetus curvatus had a total consumption of 9 individuals. In the same graph, we can see that this species, the native, was the only one able to consuming the cladocerans with large species, spines, Macrotrix tricerealis. In the graph at the bottom, when both Mesocyclus were together in the same environment, they had the highest total consumption with 11 individuals. In general, the prey species more consumed were Brachionus caliciflorus, Brachionus havanensis, and Moina macrocopa. In the other graph, the preference index showed that in separate environments, Mesocyclus pepeiensis prefer 4 species, Brachionus caliciflorus, Brachionus havanensis, Platanus parus, and Moina macrocopa, while Mesocyclus longicetus curvatus prefer fewer species, Brachionus caliciflorus, Brachionus havanensis, and Moina macrocopa. And when both species were together in the same environment, they also prefer to consume 3 species. The other one, please. Therefore, the invasive has a wider diet than the native, And this is an attribute of an invasive aquatic species. In general, the cladocerans, Alona glabra, Seriadaphnia dubia, and Daphnia pulex and Macrotristis cerealis were always avoided, regardless of whether the two copepod species were together or separated, and always the rotifers Brachionus caliciflorus and Moena macrocopa were the most preferred prey. So stomach content analysis needed to be performed, but some individuals of both mesocyclic species have already been dissected, and so far we have found that the invasive species consume the same number of prey, regardless of whether it's. On the other hand, the native species consumes 30% prey when it is present. It is in the presence of the invasive species. I think there is an interference towards the native species due to the presence of the invasive species. Thank you.
We're now open for the questions. Christian, please.
Hi, Ms. Lee. You find that when you put separated these species, 1, 8, 9, and the other one, 8, 7 prey, right? And so when you put them together, we would expect that they consume around 16 organisms, but you observe that they together consume 11. So do you think that the presence of one of these predators is affecting the feeding behavior of the other one?
Yes, I found in the analysis of content, Mesocyclos pepeyensis consume the same number of individuals, even if the presence of Mesocyclos longicetus or not. But Mesocyclos longicetus consume less individuals of prey. Mesocyclos pepeyensis were present. So for Mesocyclos, there is no interference from Mesocyclos, but Mesocyclos longicetus, they have interference when pepeyensis, the invasive species, is present. I don't know if I respond your question.
That's good. Thank you.
Okay.
Rosenberg.
Okay, thank you very much for your presentation. Actually, I have a curiosity. I was just wondering what may be happening with the zooplankton community structure in these lakes that you were sampling. Because of course that your experiment is very important to understand how the feeding behavior may influence one species or another. But I would like to know what may be happening if you have a general picture of what may be taking place in these reservoirs. That's the question that I have.
Well, yeah, I would like to have a big picture, but for that I need to work with all the community, and it will be very difficult. So now it's difficult to work with two copepods because these two copepods are very, very similar. So when they are together, sometimes it's difficult to recognize which is which. So if I put another species of plant, for example, there are another interactions, but so the idea is now to know where is the interaction with these two species who are very similar in size, for example, and in feeding behavior. But I don't know exactly if I answered your question.
No, you did answer my question. That's why I said that it was a curiosity, because I mean, if you had some information before the scopepode introduction, and then you could compare the communities before and after the introduction. So I don't know if you have this information anyway.
Add a comment, please.
Yeah, sure.
Thanks, Rosenberg. The point is that it's a very good point, and it's the next step. And in nature, it might be difficult really to find, because fortunately, we do not hope that Mesocyclops pepiensis will be spreading all over in different water bodies. But the next step could be mesocosm studies, where we add different communities and, similar to what we find in nature, and test the impact on a slightly larger scale. So that would be the next step. And then hopefully we will be able to control, but I don't think, because it looks like the species is spreading in Mexico. Over the past 10 years, it has spread over 4 different states, from South Mexico, Southern Mexico to Central Mexico. So it looks like it's on the spreading, and pepiensis has spread very widely from China all over the world now. So it looks like that it's a very, very strong invasive species. But the impact on communities on a larger scale is probably the next step with mesocosm experiments. Thanks.
Thank you very much for your answer.
Thanks.
Dr. Sarma? You are mute.
I agree. I'm sorry. I agree with Rosenberg that we need to have background information. That is why in traditional limnology, most water bodies are sampled for over a year to see the zooplankton community structure. Then you know the effect of invasive species, whether it has become exotic species, whether it has become invasive or it has no effect. So we need before and after the information, but unfortunately the species has already spread into in the water body that we have been sampling. So if we find some water bodies where these species, they are not present, then over time, if by any chance we don't have to introduce exotic species into our system to see what damage it happens, but that for that Dr. Nandini has already mentioned, for that you have a method of mesocosm. Okay, but it is a good idea to resample the water bodies that have been collected before, and now if you find any exotic species or invasive species, then you can have a comparison between and after, so to make a statistical. So it is important that even time has not passed, many water bodies still were not sampled in many countries, not only in Mexico, to have a background information and see in the future if there is any sudden invasion by another species, so you have something to compare. It's only a comment. Thank you.
Just another comment. Could be really interesting. Check it out if there are data about the feeding behavior of Mesocyclus pepiensis in its native area, and compare with your findings. It could be very interesting to see that. They compare in invaded areas, in native areas, the feeding behavior in these 2 areas. Just a comment.
Okay, yes, a good idea. Thank you.
Okay, Roger, let's move on to the next one.
Yeah, I think so.
Okay, so we invited Sol Purcell to present her manuscript.
Hi, everybody. Could you put the slides? Thank you. Well, I will present our main findings of our study. It's about the effect of remote trout introduction on food webs in lakes of the arid Patagonia. Our objective was to analyze comparatively the trophic structure and nutrients and energy pathways in shallow lakes of this strawberry plateau that you could see in the picture, evaluating water bodies with similar limnological characteristics that had been stocked with remote trout and environments in their natural fishless state. Well, to study the food webs, we base on stable isotopes analysis. We collect samples in 4 organic turbid lakes, 2 stocked, 2 fishless. We took samples from pelagic basal carbon, benthic sources, pelagic and littoral crustaceans, benthic invertebrates, and also the muscle of trout. Could you go to the another slide? Well, our first hypothesis is that trout introduction modifies the trophic structure of the community, altering the configuration of trophic relationships of a stocked lake with respect to fishless ones. To test this hypothesis, we use layman community metrics. The result of the niche area that you can see in the first plot and show substantial differences in the trophic structure between fishless and stocked lakes. These 2 groups were clearly separate and did not especially overlap. The areas you could see that show a clear shift regarding the isotope values of stocked lakes, so are more positive values of carbon and nitrogen, and it could respond to nutrients recycling of trout, and because of the organic matter mobilization from benthic reserves. It is in line with what we observed with higher concentrations of DOC, you can see in the table, and higher conductivity in the stocked lakes. Also, you could see that one, you couldn't see it because it's a bit it's small the letter, but this table summarized all the metric results. I only told you that the mean nearest neighbor distance and the standard deviation were the most informative metric about the trout effect, showing lower values on lakes with trout. This means that the organisms belong to stocked lakes were more grouped within the food webs, so there was a more uniform species distribution, which would indicate greater trophic redundancy. Well, this could be because trout and the big copepo, Parabrotea cerci, occupy similar trophic role. We go to another slide. Regarding our second hypothesis is a state that trout introduction generate change in the spatial distribution and or diet of its main prey, that's amphipods, affecting the trophic position and forcing the use of alternative carbon sources. So in the first plot, we show that the amphipods collect in pelagic and littoral zones show carbon values related to the littoral zone in the 4 analysis leg, yes, to more positive values of carbon. However, in fishless lakes, large amphipods had more negative carbon values than the small and medium size, closer to the pelagic zone. In contrast, in the stocked lakes, the carbon values of the amphipods from all sizes were clearly more enriched in carbon. Finally, you can see in the plot above, the large amphipods show shorter distance to all other compared groups, yes, particular organic matter, Daphnia, Bocchella, and Parabrotea cerci, than the medium size of amphipods, indicating that the higher dependence on pelagic reserve for the larger ones. Additionally, the larger and medium size of amphipods of stocked lakes show greater differences in carbon when contrast with the values of POM, the particular organic matter, Daphnia, and the small Bocchella, than the equivalent comparison in the fishless lake. So it's implied a great association of amphipods with the littoral zone in a stocked lake. So it could be an anti-predatory behavior. Well, in conclusion, our research reinforced the concerns about the effect of trout on primary reproductive habitat and on the survival of hooded grebe, as you can see in this picture, that is an endemic and critically endangered water-bird species. So for more information, you could see the whole presentation. Thank you.
Thank you, Sol. Now we are open to the questions.
Hi, Gustavo. You're raising your hand, so the word is yours.
Thank you. Hi, everyone. My question is more of a curiosity of my part while I was reading your work. This truth, how come she was introduced in these lakes? I mean, it was all human, right?
Yeah, well, these lakes are in this plateau that is in the arid Patagonia, and they stay in private and public lands, but the owners of the files decided to stock the trout, introduce the trout to commercials for commercial, so to gain money. I don't know. So there's a great development of the agriculture since 20 years ago to now. That's a great develop of this activity. Not only in this lake that I mentioned or area study, if not in several others in the Strawberry Plateau.
Yes, thank you. I was thinking about this while listening to your presentation, that in a ecosystem, right, a conjunction, what that is constantly diminishing its size, as you show it with the GIF, the passing of the years and the lakes reducing the size and the number of lakes in all, that some people would find it useful or obvious or for money gains, introducing a new species in these habitats that would obviously cause some harm to native ones. But I think it was just this. Thank you.
Thank you, Gustavo.
I have a question.
Yes.
Thank you. I like your study very much. I just wanted to know one thing. Does the diet of the amphipod change in the presence and absence of the fish? What does
No, it's okay. Sorry, I interrupt you.
No, what does it feed when the fish are not present and when the fish are present?
If I understand your question, we took samples and we fixed this from littoral and pelagic zone in the lake, and it's all death. That's your question?
Not littoral and pelagic, but between the lakes with and without fish.
Yes, we go to the environment and take the samples and fix it, and well, we then evaluate the stable, the isotope values.
Mm-hmm.
So all the organisms are dead. I don't know if that's your question. Sorry, I don't understand.
No, but did you do any stomach content analysis of the organisms from different lakes?
Okay, yes. In this study, we're working from other years before. So one of my director did that. They did that. So we continue. Then what they see that the more important prey was amphipods, we decided to concentrate in this particular prey, and so we complement that study of the stomach with the analysis of isotopes.
Okay, but you're talking about the fish diet content, but I'm asking about the amphipods, but it's okay. We can discuss it later. I'll write to you.
Okay, thank you, Nanindi.
More questions?
I'm sorry, Roger, but just for curiosity, the primary producers, the carbon, is different between the 4 lakes. Did you measure that or not?
We measure the particular organic matter as the base, yes, to complete all the pathways. We take particular organic matter as the first producer, in the pelagic zone, and then we scrapping the biofilm in the littoral zone of each lake with the same method. And if you see in the presentation, I couldn't say here because it's a lot of information, but you could see in the graph that the particular organic matter is more uniform during all the lakes, and the biofilm is very variable. So we couldn't use that to calculate some things. So it's something to take into account for other studies. Thank you, Bruno.
Roger, we can move on. Rosenberg has a question.
Yeah, it's a very quick question, Saul, and thank you very much for your talk. It was very interesting. I was just wondering if you had sampled zooplankton, phytoplankton communities as well. Did you do that, or was not really important for the kind of work that you were performing?
No, yes, it's really important. This is part of my PhD study. So this is one chapter about stable isotopes analysis, but I also studied the phytoplankton communities and zooplankton communities. But I don't mix yet all that information. I don't make study pay attention all of these things, but it would be really interesting. Thank you.
Okay, thank you.
Thanks, Saul. Thanks, Rosenberg. Let's move on. So we invite Ivan Saavedra Martinez to present his work.
Hello, partners. Good morning. Hi, Well, the name of the work is Population Dynamics of the Introduced Crayfish, Procambarus clarki, in the littoral of the high altitude Mexican reservoir. Procambarus clarki is a crayfish native of the northern Mexico and the southeastern United States. It has rapid growth, high resistance to environmental change, and tolerance to disease and parasites. These characteristics has given it great commercial importance. Because of this, it has been worldwide introduced in many aquatic systems, causing serious problems. Recently, has been introduced into a Tashima reservoir located in the state of Mexico. This reservoir has the category of protected natural area where the presence of endemic species has also been reported. The aim of this study was to determine change of abundance fecundity 6 radio morphometry associated to environmental variation during a year. Next, please. Well, the left graph shows the abundance. We captured total of 464 organisms. The months corresponding to the greatest abundance were May and April. The left graph shows the gonadosomatic and hepatosomatic indices. The one-way ANOVA shows significant difference in both indexes. In the gonadosomatic index, the months that differ were September, February, June, and July. In the hepatosomatic index, the months that present significant difference were May, June, and July. Reproductive season occurs between the months of June, September, and February, March. Next, please. We test a principal component analysis for morphometrics and show difference between males and females, length of the right and left calliped, and total length. Well, the Tashima reservoir has the physical and chemical characteristics necessary for the development of Procambarus clarki. Well, that's all. Okay. I'm ready for the questions.
Okay, Ivan, now we are open for the questions.
Yep.
Okay, I have a comment, as I said, for Mitsley. Could be really interesting to check it out if there are data about the population dynamics of crayfish in the north of Mexico, in the native area. and compare with these findings in the invaded areas to discuss the findings. This could be very interesting.
Okay. We don't have no data appear of this work in this part of the area, but in the north of Mexico, there are many works about the distribution and basically the population dynamics of this species. I found that in the north are many. They have many more abundance in the central part. Okay. Okay. Well, it's okay. Anyone have another question
or comment?
Or comment? Yeah.
Yes.
My English is not the best.
I'm trying.
Don't worry. It's a nice presentation. I just wanted to know what is the main mode of dispersal of these organisms? Are they normally introduced into the water bodies by humans? Or is there any natural mode of dispersal?
Now, the principal cause the distribution of this species is for activity humans, but well, it is how to say? Well, it's widely reported the damage it can be generated because of the introduction of human activities in the area where this is native. The distribution of Proponents clarky is limited by mountain systems and the aquatic systems. Yeah, but the first reason of the introduction is by activity. The failed aquaculture is the farm of these organisms.
Because I saw one map of yours which showed a very wide global distribution of the species.
Yeah.
Is this species very common as a food item, gourmet dish or something?
Proponents clarky has a general diet, and these characteristics can contribute.
I'm asking that do humans like to eat it? I mean, do we like to eat it?
Okay. In Mexico, not it's very common, but example, United States, the farming of this species is very high. In China, it's very popular aliment. So see, the people eat them, but.
Thank you, Ivan.
Okay, thanks, Doctor. Anyone have another question or comment?
I have like a curiosity. Can I ask something? You said this cray is introduced by human activities.
Yeah.
But once it is in a place, did it spread by itself? For example, in the case of my copepod, it has been introduced by human activities, different human activities. But once it is in a lake, for example, or in a river, it starts spreading by, fishes or by, I don't know, by birds because of its, with eggs or all of that. It's not like once it is in a lake, it starts spreading because other things, not just by human activities. Is this the case with your organisms, for example?
Yes, this species can move in another rivers. For example, when the lands are going to be float, these species have the capability to move in the lands, in the wetlands, and they can, well, this reason can contribute to the dispersion of the species. and another authors mention that the altitude can restrict the distribution of the species, but these species have the capacity of move by terrestrial, he can walk around with the presence of humidity. Okay, well, yes, he can do it. Sorry.
Oh, don't worry, it's okay. Thank you.
Okay. Rosenberg, do you have any question?
So I think there is no more questions.
Okay.
And thank you very much, Ivan.
Thanks guys.
It was very good to hear you, Ivan.
Okay, thank you.
And the next one is my dear old friend, Dr. Roger Mormul, or Mr. Jason, I don't know. Presenting the manuscript, Non-Native Fishes, Homogenize Native Fish Communities and Reduce Ecosystems Multifunctionality in Tropical Lakes over 16 years.
So I think I'm the last one, no?
Yeah.
Yeah, we have almost no time, so I will try to be quite fast here. So our study is about the relationship between non-native fish and native fish communities in a floodplain area. So we have long-term ecological research developed since 2000, and we take samples of many variables during this project, and now we try to evaluate this relationship because we have some changes through time in the floodplain area related to constructions of dams upstream and downstream our floodplain area. So we have through time decrease in phosphorus level and also increase of water transparency in the water of the main river in the floodplain, and this affected the adjacent environments because we have a flood pulse yearly, and this pulse connected the main river of the floodplain with the adjacent environments. So it's a common, I can say that it's a common event we have in the floodplain. But in our floodplain, we have also many non-native species because when the dam was constructed downstream the floodplain, there was an inundation of a big waterfall, and the fish that was downstream now is upstream and invaded the floodplain. So we try to visualize how this affects our fish, the native fish community. The next, Bruno, thanks. So what we found was that there is a relationship, a positive relationship between native fish and non-native fish if we analyze locally. For example, if you only take samples of the lakes and doesn't matter the time, just evaluate how one local how we can find the relationship between native species richness and non-native species richness, then we have a positive relationship. But if we consider time, then we can see that over time, this relationship is become weaker and can become negative. That it's to summarize what we found in our relationships. And it happens not only to species richness, but only to beta diversity of native species and beta diversity of non-native species. So we evaluated how the species richness increased or decreased over time, how is the relationship between native and non-native species richness, but also we evaluated how the distribution of this species in the floodplain is. So if we use a relationship between the beta diversity of native fish against the beta diversity of non-native fish, we can see over time that this relationship also changed. So here, similar to Dr. Sarma said, we don't have before and after the invasion, but we have the invasion process, and we, how can I say, we track this invasion process. And after 16 years, we can see that relationships in the beginning of the invasion now changed the environment. And we also found that not only the relationships between non-native and native fish changes over time, but also how ecosystem function over time also changed. So we found that we have environmental effects on ecosystem function. For example, changes in water transparency can affect productivity, decomposition, but also we have effect of non-native fish on the productivity and on the decomposition. That's to summarize what we found in this work. So here in this figure, we can see that we have environmental drivers, mainly variations in water level fluctuations, and also changes in water transparency and so on. This affected not only the non-native, the native fish, but also the non-native fish. This environmental change caused by the construction of dams affected negatively the native fishes, but affected positively the non-native fish. And also this changed the relationship between non-native and native fish. So the ratio between non-native and native fish increased through time. So we have decreased of natives, increased of non-natives, and this led to a change in the composition and productivity in our lakes in the floodplain. So what we put here to take home message is that the non-native fish caused a kind of homogenization of the native fish species over time. And it results in the impoverishment of the ecosystem multifunctionality, mainly productivity and decomposition. So it is our main findings here. So just to summarize the video. Okay, so we are open to questions, please.
I have one question. I was just wondering, and well, anyway, congratulations for the work. It's really nice to see this paper already published, by the way.
Thank you.
And I was just wondering if you ran structuring equation modeling, comparing the flood period with the dry period. I don't know if you did this, if it's relevant or not. So I just wondering that. And another question is if you tried to understand what was taking place with the functional structure of the fish communities, with the same type of analysis that you did. You did for taxonomic diversity and also beta diversity. I was just wondering what would be taking place with the functional richness and also functional beta diversity. I don't know if it would be possible to run with the kind of data that you have.
Yeah, it's possible. So to the first question, we didn't compare the flood period and the drought period, for example, because we used the water level as one of our drivers. So I think using the water level, we have the variation of the droughts and floods into this variable. So it is into the model. That's one of our premise. So that's why we didn't compare the flood period and the drought period. And regarding the functional, We, after prepared this manuscript related to non-native and natives, we took this example and tried to see how the functional variation occurs in these lakes, but not only visualizing the non-native and native relationship, but the whole community. And we published, it is just published in Functional Ecology now, and we have its early view, just to show that there is a huge variation also in the functional diversity and functional, how can I say, we used more than fish in this new approach. So we used invertebrates, algae, macrophytes, and we can see that independent of the trophic level, we have changes in the trophic diversity and also in the functional diversity, and these affected the functioning of the ecosystem. But we didn't compare native and non-native in this new approach, in the functional approach. It could be a nice attempt to evaluate how native and how non-native species can differ, for example, regarding the functional diversity.
Okay, thank you very much for your answer, and congratulations again for
it's
very
pleasure.
nice work.
Okay.
I would like to know what kind of interaction is there between the non-native and the native fishes. For example, they compete for food, or they are competing for space, or
yes, there are a lot of interactions. One is competition. We have some non-native species that has niche overlap with the native species, so they have similar diet, and this also affected the ratio between non-native and native. We have, for example, non-native fish species that eat, for example, larvae and eggs of native fish species. So it's also affecting reproduction, mainly the migratory fish. Also, we have predation pressure between the non-native and native species. So we have, for example, Cycla, the genera Cycla, many species of Cycla. So the cichlid fishes are visual predators, and we have the increasing transparency. So it facilitated to find prey, and most of our diversity of fish are small fish. So it's easy to the Cycla, for example, eat these small species. So it decreased, for example, small fishes, native species in macrophyte stands. So we have also a study showing that the introduction of the cichlid species decreased the number of species associated to submerged macrophytes in some areas in the reservoir and also the floodplain. So that can exemplify some of the interactions we have between the non-native and native species.
Thank you.
Welcome. Any more questions? Bruno, I think the word is yours.
Hey, now I want to thank you very much for your attention and patience. And congratulations to all the contributions, the nice contribution that we received in this thematic session. So see you tomorrow.
Bruno, I was just wondering if we could take a screenshot to post on the social media as well, if it's possible. If everybody agrees, we can take a screenshot here and post on Twitter, Instagram, Facebook, if you don't mind, of course.
Okay.
Okay, I'm going to take the screenshot now. Please smile. Think about the nice beaches here in Brazil. So I would like actually to see all of you here, but it's nice to see you here as well on Zoom. Okay, screenshot the first one, just another one, just a second. And that's it. Thank you very much. It was really nice session, and was really nice to discuss a little bit about biological invasions with all of you.
Thank you very much, all.
Yeah, thank you very much. It was a pleasure.
Thanks for the organization.
Thank you all.
See you soon. Hopefully.
Hopefully. Bom, irmã, a gente pode fechar a sala agora, não tem problema.
De boa. Tá bom,
Então, vou
tchau.
sair, tá bom? Tchau.